Okay, please note the information Dr. Morton put up for this week's lab. And then quiz for Monday will be section 9.3, pages 183 through 189. That talks about the cell cycle and a little bit of introductory information about cancer. That's your quiz for Monday. Questions? Okay, before we proceed, let's step back a little bit. If you are a bacteria, let's just say a bacteria like E. coli living in your gut, why is it really important for you as a cell to be able to regulate expression of your genes? What advantage does that do?
You don't waste your energy on processes that you don't need.
Okay, you don't waste your energy making things you don't need, and think about how the environment of that bacteria in your gut changes. Your diet isn't consistent from day to day. You're providing those bacteria with different potential food sources. So sometimes there's lactose present, sometimes there isn't, sometimes there's tryptophan present, sometimes there isn't. And so being able to regulate gene expression and save the energy of not transcribing the gene to a message and then translating it into a protein and then not using it is really, really important. For prokaryotes, most of the regulation of gene expression occurs at this level of transcription. Okay? Now, for eukaryotes, why is it important? Well, you could cite some of the same reasons, conservation of energy and things. But there's another time in an organism's life, particularly if it's multicellular, that it's really important to turn on and turn off genes. And that is during the process of growth and development and tissue specialization. So we talked a little bit about how liver cells can be different than eye cells of the lens. Well, in eukaryotes, there are all kinds of possible levels at which expression of genes can be regulated. And we've talked already about chromatin modification. So, which is expressed more readily, heterochromatin or euchromatin? Euchromatin, which is the more loosely packed DNA. Okay? What chemical modifications to chromatin can result in it being less transcriptionally active or more compacted? What can we do?
You can add a functional group.
Okay, we can add a functional group to either DNA itself or what else? To the histone proteins, okay? So if we acetylate the histones, for example, that tends to promote loosening of the DNA structure and therefore increased rate of transcription. What happens if we methylate the DNA? What does that do?
Pretty much a long-term investigation process.
Yeah, it helps kind of inactivate the DNA. It tightens up that DNA structure. We talked about bar bodies last time, for example, being that DNA that was really kind of permanently almost condensed. Okay, so those chemical modifications are really important. Now, do eukaryotes regulate gene expression at the transcriptional level? Yeah, we talked about that too. So, a little review. Here are some regions of DNA that are really important when we talk about this regulation at the transcriptional level. So basically, here are our coding regions of the gene, the exons, separated by these introns. We've got this promoter region here, and associated with that promoter region are these proximal control elements. These are a bunch of A, T's, G's, and C's. And one of the things that associates with these proximal control elements are some proteins called transcription factors. And there's certain transcription factors that every gene needs to have available in order for transcription to occur. So those general transcription factors tend to associate with these proximal control elements. That allows the polymerase to come in and then allows transcription of that gene to occur. But then, for most eukaryotic genes, we have these regions called enhancers, which even increase the rate of transcription more. And they consist of several, usually, distal control elements. What interacts with these? Other proteins, other transcription factors that we call specific transcription factors. So we looked a little bit at that molecular interaction. Whoops. And talked about how if the right factors are available, then they can interact with these distal control elements, cause the DNA to bend, bringing these regions in close proximity with one another. And then those bound activators can recruit other proteins. These were called mediator proteins, some other specific transcription factors, and allow that gene to really be transcribed very effectively. If you don't have the right activators available, then that gene really isn't going to be expressed. So that's some of the stuff we've talked about already. After transcription, there are several other opportunities to regulate gene expression. And one of the levels that we have talked about already a little bit is RNA processing. RNA processing involves removing these introns and then joining the exons together so that we have this now intact bit of information that codes for polypeptide structure. What are the molecular machines called that splice out those introns? They're called spliceosomes. Okay? And a spliceosome is a molecular machine that consists of a whole bunch of proteins and an interesting type of RNA that most people typically don't think about or even know exists, called small nuclear RNA. And these RNAs and proteins aggregate together into a structure, kind of like a ribosome almost, and that recognizes those introns and then cuts them out. And then once that has happened, then that message can be translated. Well, what we talked about was there are different ways that these introns can be spliced out. And I used the example of the human troponin gene, where we've got 5 exons separated by 4 introns. This is the primary RNA transcript. But when these introns are cut out, we can then cut and splice in different fashions so that basically the same genetic information can be used to produce 2 different messages and therefore 2 different proteins. A lot of work in this has been done on Drosophila, the fruit fly. And there's one gene in Drosophila that has enough of these exons, remember those are the coding segments, separated by introns, that one gene can produce 19,000 different membrane proteins. That's a whole lot of different combinations. Well, in Drosophila, each nerve cell, each individual nerve cell has a different membrane protein. And so this is really important in differentiation of Drosophila tissue, particularly of the nervous system. This is happening in our cells too. It's estimated that about 90% of our genes are alternatively spliced. This is one of the reasons why it's not quite as surprising as it might have seemed that we really have far less genes in the human genome that code for protein than we really thought we did. Initially, the estimate was about 100,000. Now we're down to an estimate of about 20,000 genes in the human genome. That wouldn't be possible to have that few genes if we didn't have this alternative splicing mechanism. Well, what regulates how things are spliced? Well, especially these small RNA molecules regulate that process. Spliceosomes somehow recognize when they need to splice one way and when they need to splice another way. And how they do that, I haven't a clue. Okay? There's a whole bunch of complexity of this regulation of gene expression that we don't even begin to understand. But that's another possible level of regulation. So, let's say that we make a message. Okay? We splice the pre-mRNA, join all the exons together in some order, and then we translate that into a protein. There are some other possible levels of regulation available to us now, and those involve regulation at the messenger RNA level for both degradation of the mRNA and translation of the mRNA. Messenger RNA molecules don't last forever. Every molecule in the cell has a limited lifetime. At some point, it's turned over. It's degraded, and then if you need to make it again, it's resynthesized. How long a message lasts is really an important feature of how many times it is translated into protein and how much protein is produced. So regulation of messenger RNA degradation is another area where we can regulate gene expression. Typically, eukaryotic messenger RNAs live longer than prokaryotic messenger RNAs. And one of the reasons for that is that poly-A tail that is stuck onto the end of eukaryotic messenger RNA. Don't have that in prokaryotic messenger RNA. I liken that to kind of a molecular hourglass that those messenger RNAs have. And as that poly-A tail gets successively shorter and shorter and shorter, then at some point in time the messenger RNA is no longer protected from degradation, and then it is broken down and recycled. There are also some little small RNAs called non-coding RNAs, and I'll define that in a minute, that can also signal messenger RNA to be degraded. And that's going to be really important in a minute. At the end of the period, or in about 15 minutes, I guess, we're going to see a little short segment of video on this new exciting area of regulation of gene expression called RNAi, or RNA interference. And RNAi is the result of these small RNAs, these non-coding RNAs, selectively degrading certain messages or potentially blocking their translation. So this is another place where we can regulate gene expression. Even if the message is around, there are certain things that have to happen in order for that message to be translated into protein. We need initiation factors. Those are proteins that must be present in order for the initiation complex of translation to form. We need elongation factors so that during protein synthesis, as that ribosome moves down the message and strings together amino acids, several specific proteins called elongation factors have to be available. Well, if they're not available at the right time or the right location, then translation's not going to occur. So the availability of those factors is another regulatory mechanism. And then we've again got these non-coding RNAs that can cause translation to be inhibited. So non-coding RNAs play a role in both degrading messenger RNA so it can be translated, or inhibiting the translation of an existing messenger RNA. Even once we've made a protein, many proteins that are originally translated on the ribosomes aren't functional until they are further processed in the cell. A good example is human insulin. When the human insulin gene is transcribed and translated, it forms a protein called proinsulin. And proinsulin then has to actually be chopped into half to form the active insulin protein molecule. And so if you don't properly process proinsulin into insulin, you don't get the benefits of having that particular hormone. Some of the proteins that are produced have to have sugars added to them before they're functional, glycoproteins. Some of them have to have lipids added to them, lipoproteins. So there are modifications that occur even after translation before you get the functional protein that does what it's supposed to do in the cell. And there are mechanisms available that allow regulation of that processing of protein. I guess the bottom line that I really want you to kind of cope with a little bit is that in eukaryotes, this process of gene expression regulation is really, really complicated. And it occurs on all different kinds of levels, from the big to the small. And interaction sometimes even occurs between some of these different levels. And so this is something that we're really just starting to wrap our heads around a little bit. And the more that we learn about the regulation of gene expression, the more questions we have. And the more new exciting discoveries that we can make as well. So this is a good slide that just kind of represents summaries of all of these different possibilities of where we can regulate gene expression in eukaryote. Chromatin modification, regulation of transcription, RNA processing, translation, mRNA degradation, protein processing and degradation. So all these represent opportunities for regulation of gene expression in eukaryotes. The Human Genome Project has given us a whole lot of really incredibly valuable insights about how our DNA is organized. How those 46 chromosomes, about 3 million base pairs, are put together and what that DNA does. We used to think that the organization of the genome was like this. Almost all of the DNA coded for messenger RNA, which then specified amino acid sequences for polypeptide chains. And then there was a little bit of DNA in there that coded for tRNAs and ribosomal RNAs. Okay? Well, as it turns out, only about 1.5% of our entire genome actually codes for protein coding information or messenger RNA. 1.5%. Okay? So what is the rest of that 98.5% of the DNA doing? Well, a little bit of it's tRNA and ribosomal RNA, okay? And then we later found out that some of it's introns. About 20% of our DNA we think is introns. But the old idea used to be is that basically all that other 98.5% of the DNA pretty much was this junk DNA that didn't do anything. And we were just kind of carrying it around, but it really didn't have any function. Well, some studies that have been done in the last 5 or 10 years indicate that like 75% of all of your DNA is transcribed at some point in time. But wait a minute, only 1.5% codes for messages. And even less than that for tRNA and ribosomal RNA. So what's all this other stuff doing that's being transcribed but doesn't code for protein? We call that DNA that is transcribed but doesn't code for protein non-coding DNA. And we're finding out now that non-coding DNA is incredibly important in regulation of gene expression. And that those molecules are really, really important. We used to think that really messenger RNA was the only thing really worth worrying about. Your textbook uses the analogy of the rock star. Okay, so messenger RNA was the rock star. But most rock stars need support. There are other musicians that are backups. There are songwriters. There are stagehands, etc. And the show doesn't come off unless all of those backup people help the rock star do what it's supposed to do. The non-coding RNA, which makes up most of the genome, are all these backup things. And they really help to regulate gene expression to allow the rock star mRNA to do what it really needs to do. So that non-coding RNA really plays a very important role in the regulation of gene expression. So, some of the non-coding RNA are genes for ribosomal RNA and tRNA. Some of it's introns. Some of it is this. These small nuclear RNAs that are involved in making the spliceosomes. So what's the rest of it? Well, we didn't know that until about the last decade. And one really exciting discovery that a Nobel Prize was awarded in 2006 was that many of these non-coding RNAs regulate gene expression by either degrading messages or blocking their translation. The non-coding RNAs that do that are called microRNAs and small interfering RNAs. So here's a couple of additional types of RNA. miRNA, small interfering RNA. So there's 3 definitely identified non-coding RNAs that are transcribed, but they are never translated into a protein that really regulate gene expression in very fascinating and important ways. MicroRNAs are small single-stranded RNA molecules, and they can bind to complementary messenger RNA sequences. Remember what complementary means. A pairs with U, G pairs with C. These can then either degrade the mRNA or block its translation. And if that happens, the gene isn't expressed. We think now that as many as half of our genes that are intended to express protein are regulated by these micro and small interfering RNAs, these non-coding guys. And where this is really, really important is during embryonic development and the cell specialization that occurs during embryonic development. So, here's what a transcript of an miRNA gene looks like. It's a relatively small piece of RNA, and it hydrogen bonds back on itself like a tRNA does to form all these little loops. There are enzymes called dicers which can cut out segments of these. So this is cut out from here. And then one of the 2 strands is degraded. And that miRNA then associates with some proteins. And then that complex can bind to a message that's been transcribed and cause the message to be degraded or to cause translation of that to be blocked. siRNAs are very, very similar. And as far as I'm concerned, you can consider them synonymous with microRNAs. They're made out of slightly different RNA precursors. This phenomena of inhibiting gene expression by these siRNAs is called RNA interference. And this is really cool stuff. Researchers can use this to selectively knock out genes and prevent them from being expressed. So there's a way that you can induce this happening, knocking out this gene. And then you can look at the cell where this has been done and say, what effect does that have on the phenotype of that cell? And if you can determine that, then you can correlate a particular phenotype with a particular gene. Because if you lose function of the gene, the phenotype changes. There's also some really exciting stuff that's being considered right now, where this RNAi interference can be used to treat medical conditions, particularly those that are genetically based. And it'll refer to that a little bit in a little video segment that we're going to see. Where do these come from? Why did these particular miRNA, siRNAs evolve? Well, we think that it was the evolution of a genetic defense system against certain types of virus called double-strand RNA viruses. And that the cell, once this defense system developed, kind of co-opted it and used it to regulate gene expression as well. So this is very cutting-edge, cool stuff, and it's something that you'll be hearing a lot about in the future. So let's take a look. This is a little bit of a silly video. It's a science news video. And some of the analogies it makes are a little lame. But it gives you a good idea kind of what this RNA interference is and what some of its potential applications are.
These diseases work. Alzheimer's and asthma and arthritis and cancer and HIV and all the others.
Hold on.
There's a chance. It's a real chance.
Help
that
if I
we can
put it.
treat many of these diseases much more effectively, all because surprising and so powerful. Not only are we about to know much, much more about how all these diseases work, Alzheimer's and asthma and arthritis and cancer and HIV and all the others, there's a chance, it's a real chance, that we can treat many of these diseases much more effectively, all because of this one discovery called RNAi. There's a little I at the end, which I'll explain later. You don't hear much about it. But RNAi is a really big deal. The curious thing about it is the discovery of RNAi was an accident. It was a puzzle that appeared in a petunia. It was a purple petunia. But to fully appreciate this tale, let's back up a bit. Every creature, and you know this from high school, is made from a recipe that comes from its DNA, spelled out in chemicals, A's and C's and T's and G's, inside the famous double helix. Every creature has its own DNA, different for mice, and for whales, and for flowers, but to go from a chemical recipe, A's, G's, and T's, to a real creature that squeaks, or soars through the air, or turns gloriously pink, that requires RNA. RNA is the thing that turns you from a chemical code to a real, pulsing, living creature. RNA builds life. That's big. So big that to RNA researchers like Greg Hannon, RNA is more important than DNA.
DNA really works for RNA, and proteins really work for RNA.
Would you get an argument, by the way, from somebody else?
Oh, undoubtedly, sure.
So how do you get from DNA to become a real creature? Well, let's take one of those fantastic voyages and we'll show you. We're going to find DNA and, well, we'll make it a typical cell, so we're going to have to fly in and then go off to the nucleus of the cell, which will make a beautiful castle, the headquarters. And there's the DNA, the master code, inside the nucleus. DNA, says Greg, never leaves the nucleus.
You ever meet one of those mean librarians, you know?
Yes.
Special reserve section.
The one that goes, pow.
Right, you can take the thing, you can copy, but you can't take the book, because somebody else might need it.
So if DNA is locked in the nucleus, how do we get the information out to build our creature? Well, that's what RNA does. That's from copying recipes out of the cookbook and throwing them out the window, out to the cytoplasm seed that makes up most of the cell. All those recipes floating through the air, they are RNA. And to finish up, in that sea, you see hundreds of thousands, and well, we've made them into little guys with chef hats.
Those would be ribosomes. And in your world, there are chefs who are using the recipes that are written in the RNA.
And whenever a recipe lands on a chef, whatever it is, he cooks it.
Whatever it is, he cooks it.
And each recipe is for a protein. Proteins build cells, bone cells, brain cells, organ cells. So all these chefs are basically building you. You are made of proteins. And because of RNA, we can copy, we can distribute, and we can cook up you and me. And RNA's been doing this for more than 3 billion years. But there was something spectacular about RNA that nobody knew till just a few years ago, and they learned about it, as we told you, by accident. In 1986, geneticist Rich Jorgensen was working at a biotech startup company in California. He was asked to create a spectacularly dazzling flower to attract investors.
So that we could convince venture capitalists, investors, to give us more of the green stuff, more money.
Still, back in 1986, geneticists didn't know how to work that easily with, say, roses. And so
We began with a simple plant, regular garden variety petunias. Petunias being a plant that were easy to introduce genes to in 1986.
And so they decided to create a very, very, very purple petunia. Rich knew which gene produced purple. He knew how to sneak an extra copy of that gene into the plant's DNA, the master text, to be copied by that monk-like scribe.
It will be transcribed by the monk, the same as any other gene. And he'll throw the transcript out the window into the cytoplasm, where the chef will be able to pick it up and use it.
Rich thought that if he added more purple recipes, he'd get a purpler petunia. So he did it, and he waited. And what happened?
We produced, instead, white flowers.
White flowers?
Complete opposite of what we had expected. Completely white flowers. We lost pigmentation completely. Our initial reaction was that something must have been wrong with the gene that we had engineered to introduce to the plant.
A mistake?
A mistake. So we checked everything out, and there were no mistakes that we could find.
So why didn't the petunias turn purple? What happened?
The petunia was a big puzzle. Nobody understood why, when you add an extra gene for purple, you should not get more purple, but less purple. It took a decade of brilliant scientists working on petunias and fruit flies and worms and other organisms to finally work out what was going on.
And what was going on is, quite by accident, Rich had discovered a secret inside living cells. Cells from time immemorial have had a mortal enemy called the virus. So let's imagine that the virus is a pirate ship. It lands. It then sends invaders inside the cell to shower recipes down to those cooks. But some of those recipes, you'll notice, look a little different. And what's in these recipes is not good for the cell.
No, it's decidedly not good for the cell, because the sole purpose of that virus is to make additional copies of itself, and to the point that the entire cell is filled up with this. And the cell explodes, releasing these viruses to go and then infect whatever other cells they can find.
So the theory is that long ago, cells developed a secret defense system which we will call the cop. What the cop does is when viruses invade and create showers of murderous recipes, the cop looks and thinks, some of these have a very fishy shape. It's a chemical difference which comes down to some of the viral recipes are 2 pages instead of 1, and one side is a mirror image of the other. But the point is, to the cop, there's something not right about this shape. So when they see in that shape,
They say virus. They say,
and the cop destroys the recipe. And when you say it destroys, is this, should we think like kung fu kind of thing? Is it like,
yeah, a little enzymatically, a little thermodynamics.
Enzymatically?
Enzymatic kung fu, yeah.
The cop destroys not only the oddly shaped version, whenever he sees that recipe, oddly shaped, regular shape, that recipe in any form must be destroyed to defeat the virus. And the interesting thing is, until 1998, no nobody knew that cells had this defense mechanism.
We had no idea it was there. That's what's so amazing, is this whole mechanism had been sitting there where cells were able to tell that something was very funny when they saw mirror image messages and start not just destroying the messages, but destroying anything that looked like that message. They worked out this whole defense system against viral RNA, and we then accidentally stumbled into using it.
The accident was Rich Jorgensen's purple petunia. The question, remember, was when Rich tried to make his petunia more purple, why did it turn white? Well, the answer, it turns out, was that Rich, by accident, discovered the cop. When Rich invaded the petunia cell and inserted his make more purple instructions, he didn't know it, but his purple instructions happened to have that suspicious viral shape. So when the cop saw the recipe, the cop thought, virus, and destroyed every recipe for purple in the cell.
So there is no possibility anymore of producing the purple pigment because the purple transcripts are gone.
If there are no recipes for purple, the chefs don't cook purple.
And because there's no purple pigment produced, the flowers will be white.
And that's how Rich and his petunias helped discover what we now call RNAi.
RNAi means RNA interference, because the cop is interfering with RNA messages, with the recipes of the cell.
And when scientists realized that every plant and animal cell has RNAi, a way to turn off the recipes, turn off genes, they thought, maybe we can use these cops to work for us.
Okay, Trevor?
Which brings us to Marty Russell, 78 years old. She and her husband used to spend lots of time here at their daughter's nursery.
Thank you, Rosie.
Years ago, she enjoyed doing lots of things.
Her passion was reading. She would read everything.
Golf.
Yeah.
I'd have to play golf, bridge.
But then Marty began losing her sight.
Couldn't see, and I'd probably get the peppers in with the zucchinis, and there'll be big problems then.
So she went to her doctor, who told her
You have macular degeneration.
a degenerative disease caused by too many blood vessels growing in the eye, underneath the retina.
As these blood vessels grow, they leak out fluid and blood in the center of her vision. And it's as if you're looking through a very dirty windshield, essentially.
I went home, I was just devastated.
So Marty volunteered to be a candidate for RNAi therapy, something so new she's kind of a pioneer.
She was probably one of the first to get it for any disease whatsoever, and specifically for macular degeneration. Hey, how are you?
Kaiser, how are you?
Good to see you. So this is the VIP room.
I feel very honored.
The reason Marty has so many blood vessels growing in her eye, clouding her vision, is there's probably a mistake in her DNA, a gene that produces too many recipes that say, make more blood vessels. So the chefs cook up proteins for more, and she ends up with too many blood vessels. Her doctor wants her to have fewer blood vessels. But how do you get the chefs to make fewer blood vessels?
It's pretty easy. You want to shut down a gene? Put in a copy of the gene with its mirror image. Signals the cell, better shut this thing down.
We inserted the needle after numbing her up.
So the doctors literally injected RNA recipes into Marty's eyes that said, make more blood vessels. But they made those recipes look dangerous, like viral recipes, hoping the cop in Marty's cells would leap to it and destroy lots of recipes for more blood vessels, leaving Marty with fewer blood vessels. They wanted the cop to turn off Marty's disease. Did it work?
Marty's vision has improved. It's a very promising result,
I can play bridge.
which is very
I'm not
important.
great, but, it's part of my life.
She can see flowers again.
Oh, some of them are just gorgeous.
So apparently they did trick the cop in Marty's cells to reduce vein production, although not completely.
I see the yellow. Inside is just a little cloudy, but I can see it.
There's a lot of questions still that need to be answered. This is not a treatment that is proven.
Can we deliver recipes to the right cells?
Lovely.
Does the treatment last?
That's beautiful.
All these are big questions. Still, in mice, RNAi has been effective with Huntington's disease, Lou Gehrig's disease, hepatitis, breast cancer. So says Greg, if we ever work this out in humans,
Any sort of disease that you can imagine becomes fair game.
all the disease diseases which would be helped if you shut off a gene.
Cancer, HIV, for example.
Wait, is this because you're just an RNA buff that you're saying you just listed cancer and HIV? These are famous big fat diseases.
Arthritis.
Well, stop listing them and tell me, is this a prejudice that you're telling me, or is this true? I mean, these are all candidates for this kind of therapy?
Certainly they are.
And finally, we have saved the best for last. The true power of RNAi goes even deeper than finding cures to terrible diseases. Because what RNAi does, remember, the cop's job is to turn off information, turn off genes.
The big problem of understanding, say, the human genome is you have 20,000 genes. How in the world are you supposed to know what each one does? Well, one very good way to start would be to turn off gene number 1 and see what went wrong.
So you could go through all the genes that make up a human, or for that matter, make up a petunia, and turn off each gene one at a time. If you trick the cop to turn off gene number 1, no color. So gene number 1 is involved in color production. Try gene number 2. No petals. Gene number 2 involved with petals, and so on.
You could make too many leaves, they could curl up, they could be upside down. Almost anything could happen.
But getting rid of the gene tells you what the gene does when it's working.
That's right.
The RNAi discovery is just amazing. 10 years ago, when we were sitting around talking about what would we really need to understand the human genome, we all said we would need some magic way that you could turn off any gene at will, just based on knowing its sequence. And what's happened is this discovery by scientists about RNAi has given us exactly that. It turns out that nature already had a way to turn off any gene at will.
And now, with RNAi as their key, scientists will have the means to decode every living thing, to identify the genes that allow us to grow, that allow us to move, that give us beauty and color. RNA is a modest little molecule, but what it gives us is the world. So now we go from the profound to the creepy.
One of the things that I think is interesting about this particular discovery is that usually when people think about breakthroughs in research, they really concentrate on animal research and plant molecular research in particular. They see some applications for it and they go, well, yeah, maybe you can improve crops to produce a better yield. But here's an instance where some very basic research in a plant system resulted in this brand new discovery. And this new discovery is just going to be revolutionary for understanding all genomes, including plant and human genomes, in application to all kinds of things, including human disease. So this is a good place to break. What we'll start talking about tomorrow, and really next week too, is now that we know something about how gene expression is regulated, what happens if it messes up? What happens if we get inappropriate gene expression? And the answer is, one of the possible outcomes for that is cancer. So the material we're going to talk about first has to do with the cell cycle, because cancer cells divide out of control, so you have to understand a little bit about what regulates normal cell division. And then talk a little bit about some of the characteristics of cancer, and then talk about what we've learned about molecularly what's happening to result in this alteration of gene expression. So that's what we're going next week. Okay?